Welcome to the deep dive. If you're a medical learner, especially in your first year, and you're looking to really nail down some core concepts in physiology and biochemistry, well, you're in the right place.
Absolutely. Today we're diving into blood biochemistry. We'll be hitting plasma proteins, immunity, and coagulation. Pretty crucial stuff.
Definitely crucial. Our goal here isn't just rote memorization, right? We want to connect the dots.
Exactly. We're digging into the why behind these processes, looking at the mechanisms and, importantly, the clinical side of things from the source materials. We want you to walk away with a solid conceptual framework.
Yeah, a foundation that actually sticks. Because blood, it's way more than just red cells carrying oxygen.
Oh, absolutely. It's this incredibly dynamic fluid packed with proteins that are constantly working, defending, transporting, making sure we don't, bleed out.
So let's start with the basics, the definitions. Getting the terms right is key, especially here.
Couldn't agree more. Slight differences in terminology can mean totally different things in the lab results you'll be looking at. Hashtag tag II. General introduction and analytical techniques.
Okay, so let's unpack the liquid part of the blood. You take a blood sample, spin it down, and you get this fluid. But is it plasma or serum? What's the actual difference?
Good question. It all hinges on whether the blood clotted or not. So plasma makes up, about 55–60% of your total blood volume.
Right.
You only get plasma if you prevent the clotting. So the collection tube needs an anticoagulant like heparin or maybe EDTA, citrate.
Things that stop the clotting cascade.
Exactly. When you spin that down, the liquid on top is plasma. And the key thing is it still has all the clotting factors floating around in it, including fibrinogen.
Okay, so plasma pre-clot liquid. What about serum then?
Serum is what you get if you let the blood clot first. You just draw it into a plain tube, let it sit, form a clot. Then, when you centrifuge it, the liquid that separates out is serum. And because the clotting happened, all those factors, especially fibrinogen, have been used up to make the clot.
So serum is post-clot liquid, no fibrinogen.
Precisely. And knowing that difference is vital. If you need to run coagulation tests like PT or PTT, you absolutely need plasma. But for a lot of other tests, maybe checking antibody levels, serum is often preferred.
Makes sense. And just zooming out for a second, this 4.5 to 5 liters of blood in an adult, it's doing so much, right? Respiration support, waste excretion, balancing pH, water levels, transporting hormones, drugs.
Everything. And a huge part of that workload falls on the plasma proteins. We're talking about 6 to 8 grams per deciliter concentration, collectively.
So how do we actually study these proteins? How do we separate them out? I remember something about salting out.
Yeah, the salting out process, that's kind of the old school method. You use different concentrations of salts, typically ammonium sulfate or sodium sulfate.
Right.
And by carefully adjusting how much salt you add, you can make different groups of proteins fall out of solution. It gives you rough fractions: albumin, globulins, and fibrinogen.
Simple, but maybe not super precise.
Exactly. It's historically important, gives you a basic separation, but it's pretty low resolution. For a much clearer picture, we rely on electrophoresis.
Ah, the gold standard. This is the one you see in clinical labs all the time.
It really is the workhorse. You take the plasma sample, apply it to a support medium. It used to be paper. Now it's usually agar gel or cellulose acetate. You put this in a buffer, typically Veronal buffer, at a pretty alkaline pH, around 8.6.
Not a high pH?
Because at pH 8.6, nearly all plasma proteins pick up a net negative charge. So when you apply an electric current, they all migrate towards the positive pole, the anode.
Okay, but they don't all move at the same speed, right? That's how they separate.
Exactly. They separate based mainly on their charge and size.
Yeah.
You end up with these distinct bands, typically 5 main ones.
Right, I remember these. Albumin moves fastest.
Yeah, albumin is the fastest, most abundant band. Then, moving slower, you get the globulin fractions, alpha-1, alpha-2, alpha-2 beta, and finally the slowest are the gamma globulins.
And then a machine scans this.
Yeah, a densitometer scans the gel, measures the density of each band, and plots it as a graph. That graph is the electrophoretic pattern.
Okay, now pause for a moment and think about this. For you listening, the real power here isn't just naming the bands, it's spotting when the pattern looks wrong.
Absolutely. These abnormal patterns are huge diagnostic clues. Changes in the shape or the size of these peaks can point directly towards specific disease states.
Let's link some patterns to diagnoses. What's the most dramatic one?
Probably multiple myeloma. This is a cancer of plasma cells. What you see is a very sharp, narrow, intense spike, usually in the gamma globulin region. We call it the M band, or M spike M for myeloma or monoclonal.
Because one clone of plasma cells is churning out tons of just one type of antibody.
Exactly. It overwhelms everything else in that region. Okay, what about something more common, like an infection?
Like an acute infection, what changes there?
You tend to see an increase in both the alpha-1 and alpha-2 globulin fractions. Why? Because many of the proteins that increase during inflammation, the acute phase reactants, migrate in those alpha bands.
Okay. Now, a tricky one, nephrotic syndrome. This is where the kidneys are leaking loads of protein. How does that show up?
It's quite characteristic. First, because you're losing protein in the urine, the albumin band will be significantly decreased. Albumin is relatively small, so it leaks out easily.
Right.
But interestingly, you often see a sharp increase in the alpha-2 globulin band.
Why the increase if the kidney is leaky?
Because some proteins in that alpha-2 fraction, like alpha-2 macroglobulin, are really big molecules. They're too large to easily pass through the damaged kidney filter. So as smaller proteins like albumin are lost, these larger ones get concentrated in the remaining plasma volume, making that alpha-2 peak stand out.
Ah, I see. It's a relative increase. What about immune problems?
If someone has a primary immune deficiency, meaning they can't make antibodies properly, you'll see a very diminished or sometimes almost absent gamma globulin band. Makes sense, right?
Yeah.
There most antibodies migrate.
Yeah, fewer antibodies, smaller gamma peak, and one specific genetic one.
A classic finding is a decreased alpha-1 globulin band, which strongly suggests alpha-1 antitrypsin deficiency. We'll talk more about that protein later, but the pattern points you towards it.
Amazing. So one test gives clues for cancer, infection, kidney disease, immune deficiency, genetic disorders.
It's incredibly versatile. And one final calculation labs often do from this is the albumin globulin AG ratio.
Right, comparing the amount of albumin to the total globulins.
Yep. Normally you have more albumin, so the ratio is typically somewhere between 1.2 to 1.5 to 1. A lowered AG ratio is clinically quite significant.
What does a low AG ratio tell you?
It generally means one of two main things is happening. Either you're not making enough albumin or you're making too many globulin.
Okay, low albumin production points towards.
Primarily the liver. Since albumin is made exclusively by the liver, chronic liver diseases like cirrhosis or severe hepatitis will lead to decreased synthesis. Also, severe protein malnutrition if you don't have the amino acid building blocks.
So you mentioned albumin has a long half-life.
Yes, about 20 days. This
Yeah.
is a key point for you to remember. Because it hangs around for so long, a low albumin level indicates a chronic liver problem, not necessarily an acute failure. The liver has to be struggling for a while for the levels to drop significantly.
Got it. So chronic liver disease or malnutrition can lower the ratio. What increases the globulins to lower the ratio?
Things like chronic infections, autoimmune diseases where the body is constantly producing antibodies, or, as we mentioned, multiple myeloma with its massive M protein production.
So the AG ratio is like a quick check on liver function and maybe inflammatory or immune status.
It's a really useful, inexpensive initial indicator, yeah? #TAG2, specific plasma proteins.
Okay, let's zoom in now from the groups to the individual proteins. We have to start with the big one, albumin. It's the most abundant by far. #A, albumin.
Absolutely. Albumin makes up about 60% of the total plasma protein concentration, typically running between 3.5 to 5.0 grams per deciliter.
Wow, that's a lot.
It is. And remember, synthesized only by the liver churns out about 12 grams of it every single day.
And that 20-day half-life, again, good marker for long-term liver health.
Precisely.
Yeah.
Structurally, it's a single polypeptide chain, about 585 amino acids, molecular weight around 69,000 Daltons. Relatively simple structure, but its functions are incredibly important. Let's start with the biggest one, osmotic function.
This is all about fluid balance, right? Keeping water inside the blood vessels.
Exactly. Because albumin is present in such high concentration and has a relatively low molecular weight compared to some other proteins, it exerts a lion's share, about 75 to 80% of the plasma's colloid osmotic pressure or oncotic pressure.
Yeah.
We're talking about 25 millimeters of mercury worth of pulling power.
Pulling water from where?
From the interstitial fluid, the fluid bathing the tissues, back into the capillaries. It's the main force counteracting the hydrostatic pressure that pushes fluid out of the capillaries. It's absolutely critical for maintaining blood volume.
And if albumin levels drop?
Then that osmotic pull weakens. If it falls too low, hypoproteinemia, the hydrostatic pressure wins, and fluid starts leaking out of the vessels and accumulating in the tissues. That's edema.
The classic example being the swelling in malnutrition.
Yes, the severe protein energy malnutrition state called kwashiorkor. See these children with distended bellies and swollen limbs, partly because their bodies can't synthesize enough albumin, leading to massive fluid shifts into the tissues.
So osmotic regulation is job number one. What else does albumin do? It's a transporter too, right?
It's a major transport protein. It's not super specific, more like a general taxi service for molecules that aren't very water-soluble.
Like what?
Things like free fatty acids, unconjugated bilirubin, which is toxic if it gets loose, steroid hormones, thyroid hormones, calcium, copper, and many drugs bind to albumin for transport through the bloodstream.
That binding is important, especially for bilirubin, preventing it from crossing the blood-brain barrier.
Critically important, especially in newborns, to prevent a type of brain damage called kernicterus. The albumin-bilirubin complex keeps it contained within the circulation until the liver can process it.
Okay, any other functions we should touch on?
Well, it can serve a minor nutritive function. The body can break it down for amino acids in severe starvation, but that's not its main job. And it has some buffering function.
Buffering pH.
Yes. Due to its high concentration and numerous histidine residues, it contributes to buffering capacity, but it's much less significant than the bicarbonate system in plasma overall.
Back to the clinical side, we mentioned hypoalbuminemia, albumin levels below, say, 2 GDL causing edema. What about finding albumin in the urine? Albuminuria.
Normally, very little albumin should get through the kidney filters. So finding significant amounts in the urine points to kidney damage. But what you really need to watch for, especially in patients with diabetes or hypertension, is microalbuminuria.
Micro meaning small amounts.
Exactly. Defined as persistently excreting between 30 and 300 milligrams of albumin per day. It's often the very first detectable sign that the kidneys are starting to suffer damage. Finding microalbuminuria is a major red flag. It predicts a much higher risk of progressing to more severe kidney disease later on.
So catching it early is key, and albumin itself can be used therapeutically.
Yes, concentrated albumin solutions are sometimes given intravenously in cases of severe burns or hemorrhage to rapidly boost plasma volume and osmotic pressure. Hashtag B globulins.
Okay, let's move on to the globulins. These are generally larger, more specialized proteins falling into those alpha, beta, and gamma bands. Let's start with the alpha-1 fraction and alpha-1 antitrypsin, or alpha-1 AT.
Right, alpha-1 antitrypsin, sometimes called alpha-1 antiproteinase. Its name tells you what it does. It's a serine protease inhibitor. Its main job is to neutralize powerful enzymes, particularly elastase, that get released by immune cells, like neutrophils during inflammation.
And elastase, that breaks down elastin, the stretchy stuff in tissues like the lungs.
Precisely. So alpha-1 AT acts like a shield, protecting tissues from being damaged by our own inflammatory response.
Which leads directly to its major clinical significance, emphysema.
Yes, a deficiency in alpha-1 AT is responsible for a significant portion, maybe up to 5%, of emphysema cases, particularly the type that affects the lower parts of the lungs.
How does the deficiency cause it?
If you don't have enough alpha-1 AT, the elastase released during, say, a lung infection or just from chronic irritation isn't neutralized. It runs rampant and starts digesting the elastic fibers in the alveolar walls. Over time, this destroys lung tissue, leading to the characteristic loss of elasticity and air trapping of emphysema.
And this is where smoking has a particularly devastating effect, right?
Hugely important connection. Alpha-1 AT has a critical methionine residue at position 358, which is essential for it to bind and inhibit elastase.
Yeah.
The oxidants in cigarette smoke chemically modify that methionine, oxidizing it to methionine sulfoxide. This change makes the alpha-1 AT molecule basically useless. It can no longer effectively inhibit elastase.
So even if someone has some alpha-1 AT activity, smoking just knocks it out.
Exactly. It dramatically worsens the imbalance, accelerating lung destruction, especially in individuals who already have a genetic deficiency. It's a double whammy.
And this deficiency isn't just a lung issue, it can affect the liver too.
Yes. Some genetic mutations that cause alpha-1 AT deficiency result in a misfolded protein. This abnormal protein gets stuck inside the liver cells where it's made. It can't be secreted properly. It accumulates, polymerizes, and becomes toxic to the hepatocytes, leading to liver damage, ranging from hepatitis to cirrhosis, especially in children with certain mutation types.
Okay, moving to the alpha-2 fraction. We mentioned alpha-2 macroglobulin earlier in the context of nephrotic syndrome.
Right. It's one of the largest plasma proteins. So in nephrotic syndrome, where smaller proteins leak out through the damaged kidneys, this giant molecule gets retained and relatively concentrated, causing that prominent alpha-2 peak on electrophoresis.
Next up in the alpha-2 group, haptoglobin, HP. This one's a scavenger.
It is. Haptoglobin is an acute-phase protein, so its levels rise during inflammation. Its specific job is to bind free hemoglobin that's released into the plasma when red blood cells break down outside the blood vessels, extravascular hemolysis, or sometimes even within them.
Why is free hemoglobin bad?
Free hemoglobin itself is relatively small, around 65,000 dol, and can pass through the kidney's filters. If it does, it can cause oxidative damage to the kidney tubules, and you also lose valuable iron in the urine.
So haptoglobin prevents that.
Yes. It binds tightly to the free hemoglobin, forming a much larger haptoglobin hemoglobin, HPHB complex, with a molecular weight around 155,000 dial.
And that complex is too big to filter through the kidneys.
Exactly. Instead of being lost in the urine, the HPHB complex is quickly removed from circulation by macrophages, primarily in the liver and spleen, which safely recycle the iron and break down the heme.
So clinically, if you suspect someone is breaking down red blood cells rapidly, like in hemolytic anemia?
You'd expect their haptoglobin levels to be low, or even undetectable. Why? Because the haptoglobin is being used up so quickly binding all the released hemoglobin, and the clearance of the complex is very rapid. Low haptoglobin is a strong indicator of hemolysis.
Okay, another alpha-2 globulin. Ceruloplasmin, the blue one.
That's the one. It's a copper-containing protein, giving it a distinct blue color. It binds about 90% of the copper found in plasma, holding onto it very tightly.
Functionally.
It has oxidase activity, particularly peroxidase activity. It helps oxidize ferrous iron at E2+, to ferric iron, Fe3+, which is the form that can then bind to transferrin for transport. And importantly, low levels of ceruloplasmin are characteristic of Wilson's disease, a disorder of copper metabolism.
And finally, the main iron transporter itself, transferrin, POF.
Right, transferrin is found in the beta-globulin fraction. Its primary role is to bind ferric iron, FF3+, absorbed from the gut or released from iron stores and transport it safely through the circulation to cells that need it, especially developing red blood cells in the bone marrow. Tag 3, acute phase proteins.
Let's talk about the body's coordinated response to stress, the acute phase response. What triggers this?
It's a general non-specific reaction to things like infection, injury, surgery, inflammation, even some cancers. Basically, any significant physiological stress.
And it involves changes in plasma protein levels?
Yes. Cytokines like IL-1, IL-6, TNF-alpha, released from the site of injury or inflammation, signal the liver to change its protein production priorities.
Okay.
Some proteins, the positive acute phase reactants, get synthesized much more rapidly. We've mentioned some already. Alpha-1 antitrypsin, haptoglobin, ceruloplasmin, fibrinogen, and importantly, C-reactive protein.
And others decrease.
Yes. The negative acute phase reactants, their synthesis goes down. The classic examples are albumin and transferrin. The thinking might be to conserve amino acids for making the positive reactants, or maybe to limit iron availability for bacteria by reducing transferrin.
Let's focus on the big positive one, C-reactive protein, CRP. Clinicians measure this all the time.
They do. CRP is synthesized very rapidly by the liver in response to inflammation, and its levels can shoot up hundreds or even thousands of times above normal within hours.
What does it actually do?
It plays a role in innate immunity. It can bind to phosphocholine on the surface of dead cells or some bacteria, and once bound, it can activate the classical complement pathway, effectively tagging the target for destruction by phagocytes.
So measuring CRP levels in serum is a good way to track inflammation.
Excellent way. For example, after a major surgery, you expect CRP to rise due to the tissue trauma, but then it should steadily fall back to normal over about 7 to 10 days. If instead the CRP level stays high or starts rising again after initially falling, that's a big warning sign. It suggests a complication like a post-operative infection or an abscess and tells you something's wrong and needs investigation.
And there's also a high-sensitivity version, HS-CRP.
Right, HS-CRP, high-sensitivity CRP. This test measures much lower levels of CRP, detecting low-grade chronic inflammation. It's become important because studies have shown that even slightly elevated HS-CRP levels, reflecting ongoing background inflammation, are an independent risk factor for developing coronary heart disease.
So inflammation plays a role in atherosclerosis, too.
It seems increasingly clear that it does, yes. #TagPHA, immunoglobulins, antibodies.
All right, let's shift to the stars of the adaptive immune system. The immunoglobulins, FEEDs, more commonly known as antibodies. These are the proteins that give us specific targeted immunity.
Absolutely key players. Now, first, let's clarify terminology again. We often see immunoglobulins migrating in the gamma globulin fraction during electrophoresis.
Right.
But it's important to distinguish. Immunoglobulin refers to the function of the molecule, its ability to act as an antibody. Gamma globulin refers to its physical property where it migrates on the gel. While most eggs are gamma globulins, not all gamma globulins are eggs, and some eggs can actually migrate in the beta or even alpha-2 regions.
Okay, functional term versus physical location. Got it. Let's talk structure. The basic unit is that classic Y shape.
Yep, the fundamental structure is a tetramer, meaning 4 polypeptide chains designated H2L2. That's 2 identical heavy H chains and 2 identical light L chains.
Held together how?
By numerous disulfide bonds, both linking the chains together and creating loops within the chains, as well as non-covalent interactions. And importantly, immunoglobulins are glycoproteins. They have carbohydrate chains attached, mainly to the heavy chains.
And within these chains, we have different regions, the V and C regions.
Correct. Each chain, both heavy and light, has a variable V region at its amino terminal end, the tips of the Y arms. These V regions, and text DHE, are where the amino acid sequence varies enormously between different antibody molecules.
And that variation is what determines what the antibody binds to.
Exactly. The unique shape formed by the text VHE regions coming together creates the antigen binding site. This is what gives antibodies their incredible specificity.
And the rest of the molecule.
The remainder of each chain is the constant C region, text CAGL for the light chain, and text CH2, text CH3, etc. for the heavy chain. The amino acid sequence here is much more consistent within a given class of antibody.
And the constant region determines the antibody's class and function.
Precisely. The type of heavy chain constant region defines the class G, A, MD, or E, and dictates the antibody's biological effector functions, like whether it can activate complement or cross the placenta or bind to receptors on immune cells.
Can we break down this Y-shaped molecule further? I remember something about enzymes.
Yes, the enzyme papain is classically used. It cleaves the immunoglobulin molecule at the hinge region, which is a flexible stretch in the heavy chain between the text-text EH2 domains.
What fragments do you get?
Papain digestion yields 3 pieces, 2 identical fragments called Fab fragments, fragment antigen binding. Each Fab fragment consists of 1 entire light chain and the V and text body domains of a heavy chain, basically, 1 arm of the Y, containing 1 antigen binding site.
Okay, 2 Fabs, and the third piece?
Is the FC fragment crystallizable? This is the stem of the Y, composed of the remaining constant domains of the 2 heavy chains. Text TH2 or sometimes text C3. Sometimes text EC region is what interacts with complement proteins and FC receptors on cells, mediating the antibody's downstream effects after it binds antigen.
Right. Now, the classes of immunoglobulins, there are 5, determined by the heavy chain type. Remember the mnemonic GAMDE?
GAMDE works. The heavy chains are designated gamma, alpha, delta, and epsilon, corresponding to IgG, IgA, IgM, IgD, and IgE, respectively. And remember, all classes will have either kappa or lambda light chains, but never both in the same molecule.
Let's run through them. IgG, most common, right?
Far. Makes up 75–80% of serum immunoglobulin. It's a monomer, a single Y unit. Functionally, it's the main antibody in the secondary immune response, the memory response. It's good at neutralizing toxins, viruses, bacteria, and it activates complement. But its truly unique feature.
It crosses the placenta.
Yes. IgG is the only immunoglobulin class that can be actively transported across the placenta from mother to fetus. This provides crucial passive immunity to the newborn for the first few months of life, protecting them while their own immune system matures. Hugely important.
Okay, next, IgA, the secretion specialist.
Exactly. IgA is found as a monomer in serum, but its main role is in body secretions. There, it usually exists as a dimer. 2 IgA units linked by a J chain and also associated with a secretory component, which protects it from enzymatic digestion.
Where do we find it?
In saliva, tears, breast milk, especially colostrum, sweat, and secretions lining the respiratory and gastrointestinal tracts. Its job is mucosal immunity. It binds to pathogens on body surfaces, preventing them from entering tissues. First line of defense at portals of entry.
IgM, the big one.
The giant. IgM exists as a pentamer, 5 Y-shaped units joined together by a J chain, making it the largest immunoglobulin by far.
Right.
Because it's so big, it's mostly confined to the bloodstream.
And its main role.
IgM is the first antibody produced during a primary immune response when the body encounters an antigen for the first time. Its pentameric structure gives it 10 potential antigen binding sites. Though usually only 5 bind effectively due to steric hindrance, making it extremely efficient at agglutinating clumping pathogens and activating the complement system. It's a rapid responder.
IgD. What about this one? It seems less well-defined.
It is. IgD is a monomer, found in very low concentrations in serum. Its primary known function is as an antigen receptor on the surface of mature B lymphocytes. along with IgM. It likely plays a role in B-cell activation, but its soluble form seems to have limited function.
And finally, IgE, the allergy antibody.
That's its claim to fame. IgE is also a monomer, present in absolutely minute concentrations in serum in healthy individuals. However, its levels can rise dramatically in people with allergic diseases and during parasitic worm infections.
How does it cause allergies?
IgE has a unique property. Its Fc region binds with extremely high affinity to Fc receptors on mast cells and basophils. When an allergen, like pollen or dust mite protein, cross-links IgE molecules bound to a mast cell, it triggers the cell to degranulate, releasing potent inflammatory mediators like histamine. That causes the immediate symptoms of allergy: itching, sneezing, wheezing.
Okay, that covers the classes. But how do we generate so many different antibodies? We encounter millions of potential antigens, but we don't have millions of antibody genes, right? This is antibody diversity.
It's a fascinating biological puzzle solved with incredible genetic elegance.
Ah.
We achieve this vast repertoire using essentially two main mechanisms. First, combinatorial diversity through gene rearrangement.
Meaning mixing and matching gene segments.
Exactly. For the heavy chain, there are multiple different gene segments encoding parts of the variable region. V, variable, D, diversity, and J, stranding segments. For the light chain, there are V and J segments. During the development of each individual B cell, the DNA in its immunoglobulin loci undergoes a unique rearrangement process called VDJ recombination. The cell randomly selects one V, one D for heavy chain, and one J segment and splices them together. The different combinations possible just from this shuffling generate millions of different antigen binding sites before the B cell ever sees an antigen. Plus, there's randomness in how the segments are joined, junctional diversity, adding even more variation.
So that creates the initial pool. What's the second mechanism?
Somatic hypermutation. After a B cell is activated by an antigen and starts proliferating, the genes encoding the variable regions of its antibody undergo a process of accelerated, targeted mutation.
Mutations happen after antigen exposure.
Yes. This introduces small changes into the antigen binding site. Some of these mutations might make the antibody bind the antigen even better. B cells producing these higher affinity antibodies are then preferentially selected to survive and proliferate a process called affinity maturation.
Wow. So recombination gives breadth and mutation gives fine-tuning.
That's a great way to put it. These two processes together allow our immune system to generate a seemingly limitless array of antibodies from a limited set of genes.
Let's connect this back to disease. When this antibody production system goes wrong, specifically when one plasma cell clone goes rogue, we get multiple myeloma.
Right. Multiple myeloma is fundamentally a cancer of plasma cells. A single clone of plasma cells in the bone marrow starts proliferating uncontrollably.
And these cancerous cells produce antibodies.
They produce huge amounts of a single type of abnormal monoclonal immunoglobulin, often called the M protein or paraprotein. Most commonly, it's an abnormal IgG, about 75% of cases. It can be IgA or, more rarely, others.
What are the consequences of churning out so much of one abnormal antibody?
Several major problems. Firstly, the overproduction of this useless M protein often suppresses the production of all the normal functional immunoglobulins. This leads to a state of depressed immunity, making patients highly susceptible to recurrent bacterial infections like pneumonia and urinary tract infections. That's often a major cause of illness and death.
And we detect this M protein using electrophoresis.
Yes, the plasma electrophoretic pattern is key for diagnosis. As we discussed, you see that characteristic sharp, narrow M band, or spike, usually sitting somewhere between the beta and gamma globulin regions, often dominating the pattern and obscuring the normal diffuse smear of polyclonal gamma globulins.
What about Bence-Jones proteins? What are they?
In many myeloma patients, the malignant plasma cells don't just produce intact immunoglobulins, they also produce an excess of either the kappa or lambda light chains. These free light chains are small enough to get filtered by the kidneys. When they appear in the urine, we call them Bence-Jones proteins. They're found in the urine of maybe 20% or more of myeloma patients. And they're not harmless. They can precipitate in the renal tubules and cause significant kidney damage, sometimes leading to renal failure. Myeloma kidney.
How do we detect them? Is it just urine electrophoresis?
That's the best test today.
Yeah.
Electrophoresis and immunofixation of a concentrated urine sample can specifically identify monoclonal-free light chains. But historically, there was a very famous, almost bizarre classical heat test.
Not the one with the weird solubility?
Exactly. You acidify the urine sample and gently heat it. Bence-Jones proteins have the unique property of precipitating out of solution between 40 and 50 degrees C. Then, if you continue heating towards boiling point, 100 degrees C, they actually redissolve. And then, as the urine cools back down to around 70 degrees C, they reappear. This heating and cooling behavior is highly specific for Benz-Jones proteins.
Wow, that's quite a specific property. Hashtag tag tag Z. Blood clotting. Coagulation. Okay, last major topic. The body's emergency repair system, blood clotting, or hemostasis. This is absolutely vital to prevent blood loss after injury.
It's a critical defense mechanism. It involves a complex cascade of reactions engaging nearly 20 different factors, mostly glycoproteins synthesized by the liver.
And those Roman numerals for the factors, they're not in order of reaction, are they?
No, frustratingly, they're numbered roughly in the order of their discovery. And remember, the active form of a factor is usually denoted with the subscript A, like factor X becoming factors N.
The whole process is usually described as starting with two main pathways that then merge, right?
Correct. We talk about the extrinsic pathway and the intrinsic pathway, both ultimately leading to the activation of factor X, which kicks off the final common pathway.
Let's start with the extrinsic pathway. Why extrinsic?
Because the key thrust comes from outside the blood itself. When tissue is damaged, a protein called tissue factor, factor 3, which is normally present on cells underneath the endothelium, gets exposed to the blood.
Ah, okay.
Tissue factor then binds to factor 7, which is in the blood, and this complex rapidly activates factor X. The extrinsic pathway is considered the primary initiator of clotting in vivo. It's very fast and gives you that initial burst of factors A needed to get things going.
Okay, so extrinsic tissue injury trigger, fast activation of X. What about the intrinsic pathway?
The intrinsic pathway involves factors that are all normally present within the bloodstream, hence intrinsic. These include factors 12, telom, 9X, and 8.
What kicks it off?
It's traditionally thought to be activated when blood comes into contact with a negatively charged surface, like the exposed collagen fibers in a damaged blood vessel wall. This sets off a cascade. Factor 12 activates X11, which activates 9X. Then, activated factor 9 forms a complex with its cofactor, activated factor 8, on a phospholipid surface, like platelets, with calcium ions. This complex, called tinase, is extremely potent at activating factor X.
So intrinsic contact activation, slower, but maybe amplifies the process.
Exactly. While the extrinsic pathway starts it, the intrinsic pathway significantly amplifies the generation of factors A.
Okay, so both pathways lead to factors A. I mean, now we're in the final common pathway. What's the next crucial step?
Factors A's main job is to convert prothrombin, factor 2, into its active form, thrombin, factor 5. But it doesn't do this alone.
It needs help.
Yes. It forms another critical complex called the prothrombinase complex. This assembles on the surface of activated platelets and requires factor Z, its cofactor, factor V, activated factor V, platelet phospholipids, and absolutely essential calcium ions, Ca2+. This complex is what efficiently generates thrombin.
And thrombin, that's the real workhorse enzyme here.
Thrombin is central to everything. Its primary job is to convert the soluble plasma protein fibrinogen, factor 1, into insoluble fibrin.
How does it do that?
Fibrinogen is a long molecule. Thrombin acts like molecular scissors, cleaving off small peptide fragments called fibrinopeptides from the ends of fibrinogen. This exposes sticky binding sites on the remaining molecule, which is now called a fibrin monomer.
And these monomers stick together.
They spontaneously polymerize, sticking end-to-end and side-to-side through non-covalent interactions, forming a loose meshwork of the soft clot.
But that's not strong enough, right?
Correct. For a stable clot, it needs reinforcement. This is where factor 30, fibrin stabilizing factor, comes in. Factor 30 is also activated by thrombin. Factor 30 then catalyzes the formation of strong, covalent crosslinks between adjacent fibrin monomers, specifically between glutamine and lysine residues. This turns the soft, flimsy clot into a tough, stable, insoluble hard clot or fibrin mesh.
Wow, quite a cascade. Given how powerful this is, we need ways to control it too, anticoagulants.
Absolutely. There are natural inhibitors. A key one is antithrombin III, AT3, and it slowly inactivates thrombin and factor Z, among others.
And that's where heparin comes in.
Yes. Heparin, the drug, which is a type of naturally occurring complex carbohydrate, a heteropolysaccharide, works by binding to antithrombin III and dramatically accelerating its ability to inhibit thrombin, AIAA, and factors A, as well as ISC, IAA, and ThreadM. It's a potent anticoagulant used therapeutically.
What about things we use in blood tubes?
Things like oxalate, fluoride, EDTA, and citrate work simply by binding up or chelating the calcium ions, Ca2+. Since calcium is essential at several steps, like for the prothrombinase and tinase complexes, removing it stops the cascade cold. That's why blood collected in tubes with these doesn't clot.
And the common oral anticoagulant, Warfarin, how does that work? It's different, right?
Very different mechanism, and crucial to understand. Warfarin interferes with the synthesis of functional clotting factors in the liver. Specifically, it inhibits the action of vitamin K.
Vitamin K is needed for.
Vitamin K is required as a cofactor for an enzyme that performs a critical post-translational modification on several clotting factors.
Yeah.
Factors 2, prothrombin, 7, I, and X. This modification is called gamma carboxylation. It adds an extra carboxyl group onto certain glutamic acid residues in these proteins.
Why is that extra carboxyl group important?
Because it creates negatively charged sites that allow these factors to bind calcium ions. And binding calcium is necessary for them to anchor properly onto phospholipid surfaces, like platelets, where the clotting complexes assemble.
So warfarin blocks vitamin K recycling, preventing this carboxylation.
Which means the liver produces factors 2, 7IX, and X that are structurally intact but biologically inactive because they can't bind calcium or participate effectively in the cascade. That's how warfarin prevents clots from forming.
Clever. Okay, so we form the clot. Eventually, we need to get rid of it. That's fibrinolysis.
Right, clot dissolution. The main enzyme responsible for breaking down fibrin is plasmin.
Is plasmin always active?
No, it circulates as an inactive precursor called plasminogen, which is made mainly in the kidney. Plasminogen gets incorporated into the fibrin clot as it forms.
How does it get activated?
By plasminogen activators. The most important one released from endothelial cells near the clot is tissue plasminogen activator, TPA. Urokinase is another activator. When TPA converts plasminogen to plasmin within the clot, the plasmin starts chopping up the fibrin mesh, breaking down the clot from the inside out.
And we use TPA as a drug too.
Yes, recombinant TPA is a clot-busting drug used therapeutically in situations like acute ischemic stroke or heart attack to dissolve the pathological clot causing the blockage.
Finally, let's touch on abnormalities in blood clotting. The most famous are the hemophilias.
Hemophilia A, classical hemophilia, is the most common severe inherited bleeding disorder. It's X-linked, so primarily affects males. It's caused by a deficiency or defect in factor VIII.
And hemophilia B.
Hemophilia B, Christmas disease, is also X-linked and clinically very similar to hemophilia A, but it's caused by a deficiency in factor IX. Both result in impaired ability to form clots via the intrinsic pathway, leading to prolonged bleeding, especially into joints and muscles.
What about von Willebrand's disease?
This is actually the most common inherited bleeding disorder overall, though often milder than hemophilia. It's caused by a deficiency or defect in von Willebrand factor, VWF. VWF has two main roles. It helps platelets stick to damaged endothelium, platelet adhesion, and it acts as a carrier protein that stabilizes factor VIII in the circulation. So defects can cause problems with both platelet function and the intrinsic clotting cascade.
Okay, that covers a huge amount of ground proteins, immunity, clotting.
It really does. And you can see how interconnected it all is, can't you? Liver function is critical for albumin and most clotting factors. The immune system relies on those complex immunoglobulins. Haptoglobin protects the kidneys during hemolysis. These plasma components are constantly interacting.
Absolutely. They're real-time indicators of what's happening systemically. Let's quickly recap some key clinical takeaways or mechanisms for everyone listening.
Good idea.
1. Remember that lowered AG ratio often points to chronic liver issues because albumin hangs around for about 20 days. Acute liver failure might not show up in albumin levels immediately.
Right. 2. The direct hit from smoking, oxidizing that specific methionine and alpha-1 antitrypsin, is the biochemical reason it so drastically worsens emphysema, especially in deficient individuals.
3. The genetic magic of VDJ recombination and somatic hypermutation is how you generate that incredible antibody diversity from a limited gene pool.
And 4. Warfarin's mechanism blocking the vitamin K-dependent gamma carboxylation needed to make factors 2, 7, 9X and functional. It's about blocking synthesis of active factors.
Perfect. Quick memory quiz.
Let's do it.
Question 1. We know IgG crosses the placenta, but think about IgM. Apart from being the first responder, what structural feature makes IgM exceptionally good at activating the complement system?
Think about its shape.
It's a pentamer. Having those 5 Fc regions clustered together makes it really easy for the first complement protein, C1q, to bind and kick off the cascade.
Nice one. Okay, question 2. The prothrombinase complex is vital for converting prothrombin to thrombin. It needs 4 key things assembled on the platelet surface. Can you name 3 of them?
Okay, definitely need the enzyme, factor Z, and its helper, the cofactor, factor V, And they need a surface, so phospholipids. Oh, and the crucial ion, calcium.
Got all four. Excellent. Factors A, factor Va, phospholipids, and calcium.
Fantastic. Okay, wrapping up. Here's a final thought to chew on. We talked about how specific protein deficiencies cause specific organ damage, alpha-1 deficiency hitting the lungs, haptoglobin loss stressing the kidneys. Now consider a protein with a really broad essential role, like transferrin, the iron transporter.
Uh-huh.
If you had a hypothetical complete, immediate knockout of the transferrin gene, zero iron transport possible, how would the speed and scope of systemic failure compare to, say, lacking alpha-1 Which condition leads to multi-organ failure faster? Think about daily essential transport versus defense against occasional threats.
That's a good one to ponder. Really highlights the different timescales and dependencies within the system.
Exactly. Well, great job working through all this. You've just tackled some absolutely fundamental concepts in plasma proteins, immunity, and coagulation. That's a really strong base for your medical studies.
Definitely. Keep reviewing, keep connecting the concepts, and you'll build a solid understanding.
We'll see you next time on The Deep Dive.